Excuse.
Oh, I thought you said record. Do you want me to stop? Okay. All right, let's just get started. So you're all here for the VCF Plink workshop. And before we dive in, well, I should say, first of all, I'm going to be toggling between screens a little bit. So if anyone on Zoom can't hear me, can't see what's going on, it's going to be hard for me to see in the comments if you have a question. So please don't hesitate to holler. Okay. So just a little bit about me. I am, I don't know why that's animated. I am a postdoc in biological anthropology. My background's in human population genetics, so I use Plink a lot. I mostly use it to look at adaptation to high altitude in two different populations, the Sherpa and the Peruvian Quechua. But I also have a background looking at novel genetic variation related to diet and health more broadly. And as part of like my PhD dissertation, I actually worked on a lot of different organisms. So I worked in Arabidopsis, I worked with insects and mice. So if any of you happen to work with other organisms and have questions, holler. Okay. So I thought it'd be nice just to briefly get like a feel for where everyone's sitting in this class. So just tell me your name if you want, your affiliation, and I mostly want to know what you want to learn out of this class so that if there's anything I can adjust for tomorrow or the next day, I can do that. And since it's the day after Halloween, why don't you say if you wore a costume, what your costume was, or if you've ever worn a favorite costume, okay? So why don't we do in person first? Who wants to go first? You go first? Okay.
I'm Serena, I'm a graduate student.
Serena. Okay, Serena.
And I'm not too familiar with population genetics. I think I've worked with it in like a class setting before. But
Okay.
I really wanted to take this workshop to get like
More familiar.
Coding experience, I guess. Intermediate, I've worked with like Python and R before. So,
Oh, you have. Great,
so
great.
hopefully that'll help with this workshop.
That will, definitely.
I didn't wear a costume yesterday because I had a nose up.
Oh no, what horrible timing. Oh, bummer. Okay, well, maybe next year. Okay, how about you?
Hi, my name is Chile. I am a graduate student as well. I studied bioinformatics program, and my interest is population genetics, really.
Okay.
So I'm part of the START program, which is a clinical pathway for fellows at UCLA to go through their research training. I'm in the cardiology program.
Oh, and, nice. And wait, I'm sorry, what was your name?
Tunde.
Tunde?
T-U-N-D-E.
T-U-N-D-E. Tunde.
That's correct.
Okay.
And I'm mostly interested in exploring genetic determinants of clinical outcomes within cardiovascular space.
Awesome.
No prior experience with genetic code.
Okay. Well, you will get some.
Yeah, looking forward to it.
Great. Okay, how about on Zoom? Kristen, I'm just going to go in the order I see people's names, but Kristen, I know you're at your visa appointment, I think. So
or passport, yeah.
Passport, sorry.
It's in a few minutes, so I'm outside, but I am like Tunde in the STAR program in the bioinformatics PhD program and have a little experience with Plink, but hoping to get a lot more with this class and moderate amount of coding experience.
Awesome. Okay, how about Asha? Asha? Tell me how to pronounce your name too. Asha Pathak. Am I saying that correctly?
Oh, I see something in the chat.
In the chat, okay. Maybe, let's do this. Ah, gotcha. Okay. Yeah, sorry. It's hard for me to see the chat when my screen is up. Okay, and then I see, okay, Asha and CNV, and then, so Asha says, hi, I study epigenetic modifications in pituitary tumors. Very cool. Sorry, my microphone isn't working. I would like to learn more about BCF and CNV. I have some experience in R. Great. Okay, and then I see Julie Ann says, I want to access and work with genomic data from Data Discovery Mart. Never worked with Plink, intermediate experience in R. Costume was gene editing. Nice, that's cool. Rip genes, very, very cool. Okay, how about Meryl Vandenberg?
I like that.
Hi, can you hear me?
Good morning.
Hi, good morning. I'm joining from Switzerland, so it's evening here.
Oh, good night. Do you speak
Yeah.
German, French, or
So
English?
I'm originally from the Netherlands, so I speak Dutch, but here in Switzerland we speak German. They're in Zurich.
Nice, very cool. Well, good evening then.
Yeah, good evening.
Do you want to,
I'm not familiar with the genetics, or very little only. I'm just a beginner in the field. We're working on lab automation, and I'm trying to learn more about population genetics.
awesome. Okay, great. You will learn more. I'm trying to think who else is left. Emma, I think.
Hello, can you hear me?
Yes, I can. Good morning.
Good morning, thank you. I'm Emma. I'm a research assistant working on GWAS project for psychiatric diseases, including ASD. And I am mostly working on Plink and VCF files, but I have basic knowledge for now. And I feel a little difficulty for dealing with VCF files, so I would like to know more, like better handle the VCF files in this class.
Okay.
Mhm.
You will get it. Excellent. And then I have Sarfaras. Am I saying that correctly?
Yeah, good morning.
Good
My name is
morning.
Sarfaraz. I work at Cedars as a bioinformatician, and I recently started in this role in the it's the IBD group, we call it here, IBIRI, which is inflammatory bowel disease research. And we focus on the translational genomics part, where we are interested in the effects of genetic variation on IBD group of diseases.
Interesting.
And yeah, and I was just recently, like I said, I recently started this and I was introduced to Plink on this job. So I'm just trying to learn more about it and get familiar with it.
Awesome. Very cool. And then I think Dan, are you taking the class or?
Yes, I am.
Oh, nice.
Good morning.
Good morning.
Yeah, so I'm a postdoc at UCLA. So my research basically involves with the multi-omics data integration. I do have some experience in mapping stuff. So I do have experience in PCF files, but I don't have any experience with Plink. And for the costume for yesterday, I didn't wear any costumes. It was typical day of life for postdoc.
Yeah.
Yes.
Working late. Yeah.
Yeah, pretty much. Yeah, nice to meeting you all.
Okay. So I think Jijun, am I saying that correctly?
Yes. Hi.
Good
Hi,
morning.
hi, Casey. Hi, everyone. Good morning. My name is Jijin, and everybody here call me JJ. That is more easier for you guys. So I'm a postdoc in the Department of Anesthesiology currently, and I'm working on cardiovascular disease. So I have been previously getting involved to using bioinformatic tools to study the heart failure, and with or without the metabolic stress. And also I'm working on another project is related to cardiomyocyte maturation using stem cell derived cardiomyocytes. So I would like to learn more about this workshop and working on learn more genetic tools. So I'm very happy to have can be joined here.
Awesome, very cool. And I think that's it. Did I miss anyone? Okay, silence is confirmation. I'm gonna take it. Okay. So I just wanted, like I said before, if anyone missed my comment before, I'm gonna be toggling between screens so I can demonstrate some things. So on Zoom, if you have a question, it is hard for me to see the chat. So maybe just put up the little emoticon hand or something if you can't talk. But so this is kind of the general format we're gonna do. I'm gonna really keep the lecturing and background to a minimum as much as I can, because I want everyone to get like a lot of hands-on exercises. I want you to walk away from this workshop being comfortable with Plink and being able to look things up on your own and apply it in whatever way you need to for your own research. But for the first day, we are going to be doing something that's fundamental to any project with VCF files, which is quality control. So that is why I had you download a few of our data files. A couple are quite big, so they take a while to download if you haven't done so already. And for the second part of this workshop, we're going to be doing QC of those data files and practicing some file manipulation with them as well. And then tomorrow, we're going to be looking at population stratification. So we'll learn how to do a PCA, we'll learn how to make an admixture plot, and we'll prune for linkage disequilibrium and minor allele frequency to kind of reduce the amount of markers that you're building these plots with. So in my email, I said it would be really good for you to download R, or if you're more comfortable with Python, that also works, because for day 2 and day 3, we're going to be making a lot of plots, and Plink doesn't produce plots. So you need some sort of other program to make your figures, basically. And then day 3, we will be doing case control association. So this will be really good for you who are looking to learn more medical type of analyses with VCF files. And we'll also be doing GWA, so making a Manhattan plot, logistic and linear regressions, that sort of thing. If we have time, we will do copy number variants. I'm not sure how the timing will go. It kind of depends on everyone in this class, but I know, sorry, I forgot who. I know one of you wanted copy number variants, so I will include the code for that if we do not get to it, and I can answer any questions. So if you look online at the Google Drive in the email I sent, there are folders for day 1, day 2, day 3, and at the end of each day, I'll upload the PowerPoint so you can review it and go over it if you want, but I'm not gonna post the PowerPoint until we've done the class, because I want you to try the take-home exercises on your own. Like, it's really painful, but the best way to learn is to just go for it, right? There's not going to be a grade with these take-home exercises, so it's really for your own benefit to try and learn these concepts. Okay, so before we get into the hands-on exercises, I'm just going to go over kind of the basics of a VCF file if you're unfamiliar or you've never opened one before. So VCF stands for variant call format, and it's basically a text file that can be used for storing marker data, genotype data. However, it does not store covariate data, which we'll get into later. But if your text file is pretty small, you can open it in a text editor like the ones I suggested, BBEdit, Notepad. You could even open it in Excel, which we'll be doing because our data sets are small. But if they're really big, you can't open them, right? You'll have to do commands to kind of get a glimpse at the data and manipulate it that way. And this format was developed by the 1,000 Genomes Project. So is anyone already familiar with this project? For those on Zoom, maybe like a little hand emoji. You see one? No? Okay, so this is a project that, oh yeah, Dan, of course. So this is a project that basically set out to do what the name suggests, right? They wanted to do genome-wide sequencing of populations around the world, but I highly recommend this resource, even if you're working in medical fields, because sometimes you could be working with data sets that are really small and you need to reference them against a larger cohort. So this is public, free information. You can always download these files and merge it with your data set to have like more meaningful information on population and that sort of thing. So there's lots of different types of genetic variants, transitions, right? So where the chemical structure stays the same, adenine to guanine, or cytosine to thymine, or transversion where they switch. There's also indels, insertions, and deletions. And then there's lots of different structural variants that you'll see in VCF files, right? You could see duplications, copy number variations sometimes, depending on where you get your data from. So it's good to be mindful of these different types of variants because they're not always just single alleles in your file. And then the two primary uses for a VCF, and if anyone uses it for something else, feel free to chime in. But what I thought of is one, healthcare, right? So you might be looking for rare diseases, you might be looking in a family for screening for different genetic variants, you might be running a clinical study, or you might have a patient that might or might not have a tumor, right? So you need to match it against a reference. They're also frequently used in population genetics, which is what I do. So you can look at ancestry of individuals, you can look at migration patterns, phylogenetics. In other species, you can use VCF files to look at different species classifications, both living and extinct. And when you actually open a VCF file, it'll have three main parts. So the first part is these kind of meta information lines, and they'll have multiple hashtags, and there could be as many as like 3 lines to, I don't know, 10 lines, right? So that is just kind of the information associated with the sequencing, associated with the organization of the file, maybe how the variants were called in what program. And then after that, you'll have, let me see when I move this, great. You'll have this header line that starts here with Chrome. And then after that, you'll have all your data, right? So each row is going to be a different variant. And then starting here, after the format column, you're going to have your individuals, okay? And then if you ever see a dot like this, that means missing data. Okay. So, oh yeah, go ahead.
Just can you go over that again?
Yeah.
The formatting.
Oh, so we're gonna, do you mean like the different columns?
Yeah.
Yeah, we're about to do that.
Okay.
No worries. Yeah. No, I love questions. So believe me, like I hate talking at a room, so holler. So I think based on everyone's background, right, this should be pretty familiar terms, hopefully, but chrome stands for chromosome. Position is the base pair position of the variant, and then the ID is the actual variant list. And here it's missing because of how it was called, but I'll show you some examples where it's present, and it just depends on the program you used to call your variants, and the reference you were using, right? So the position and the ID can sometimes vary depending on those factors. And then if we move on to the other columns, we have first our reference allele, and then we have our alternative allele. And then after that, we have our quality score. And this quality score is for basically the variant calling score. So I'll go into that later, but it basically tells you the confidence that this reference allele and alternative allele are correct. And it's called a Phred scale, Phred, and it's out of 30 usually, but this one's out of 100, which is weird. Okay, so in this example, right, we have some of the variants that I went over. So we have a transversion, right? It goes from a pyrimidine to a purine. Hopefully I got that right. And then it goes a deletion, right? We lose that C allele. And then here at this particular variant site, we have an insertion. And then sometimes if there's multiple alternative alleles, they'll be separated by a colon, but sometimes the program, especially Plink, can't handle that. So you have to filter them out, right? It's really important to pretty much keep it pretty clean and only have one option in each column. And then, like I said, so the filter basically tells you if it passed or not. So here, basically, FA means it failed filter A. So whatever that particular variant calling program was, it failed the first filter. And then here, this will tell you which variants failed. So VT is variant type and AC is allele count. So this tells you that 3 genotypes failed at this site, which is pretty unusual. Hopefully, when you get your VCF files, they're pretty good quality. Okay, so that being said, what do you notice about this VCF in comparison to the other example VCF? The ID, yeah, so the ID column now has information, perfect, yeah, so that's a slight difference. Anything else? How about on Zoom?
Quality value is empty for some
Yeah, the
reason.
quality and the filter value is empty, absolutely, it's missing data. That's a big one. Yep. And then there's one other thing. I will go back. So does anyone notice what type of chromosome this is? What's MT? Mitochondria.
Mitochondria.
Yeah, perfect. So we're only gonna have one allele information, right? Because it's inherited from the mother, versus here, we have biallelic information, which is more typical, I would say, of a VCF. And then one important thing to keep in mind is when you open a VCF, these dashes will indicate whether your data is phased or unphased. So does anyone know what phased or unphased implies? On Zoom, any thoughts? Wrong answers are accepted if you want to shout them out. Don't be scared.
Whether or not they know if it comes from the maternal or paternal allele, or if it's all mixed together and you can't tell.
That is a very good guess. You're close, but not quite there, but very excellent guess. Yeah. Anyone else have a thought as to what that could mean? No? Okay, so phased means the haplotype data has been inferred, right? So it basically tells you whether or not, so a haplotype is genetic variants that tend to be inherited together. So especially for population genetics, it's really important to know haplotype data when you start to run different statistics to look at different populations. So here, when you look at this BCF file, this genotype data is not phased because that dash is slanted and not straight. So that means that if you need to run a statistic that requires haplotype data, you will have to phase this BCF using a reference. Okay, and then as you pointed out, it is blank. Why could this be blank? So this particular VCF is one of mine from my research, and the reason it's blank has to do with how VCF file is prepared, right? So basically what happens is you get the sample from your participant, it's sequenced, it results in this FASTQ raw reads, right? After it's been sequenced, you do quality control on that, and then you align it to a genome. And then after you align it to a genome, as you can probably imagine, there are a ton of different programs for calling variants, right? You can pay a lot of money and have it done with like Illumina. They have their own in-house program. There's a lot of free programs out there where you can do it yourself, and they're all slightly different. The most common one is called GATCA, and it was developed by the Broad Institute, which is at Harvard. So they're kind of, I don't know, the typical standard of what most people do if you're ever curious as to a pipeline. And they do this quality control check after variant calling, which is called variant quality score recalibration, so VQSR. And the reason I bring this up is basically, if you are in population genetics, you are trying to look for novel variants. And this could also be the case in medical studies, right? You're trying to find a rare variant that's not in the majority of the population. So when you perform this quality score recalibration, this aligns it to a reference and common variants. So it could actually be filtering out novel variants. So depending on what your research is doing, you actually might want to skip this step. And another caveat that I bring up is it's really important to know where your VCF data came from, because if you're not looking at a white European population, sometimes the reference you're aligning it to is a white European population. So you're gonna lose novel variation in your particular cohort, right? The 1,000 genomes reference is a mixture of different people from different populations, so it's pretty good. But if you look at genetic studies overall, it's overwhelmingly white European, right? So only in the last 4 years, I think there's like a tiny percentage that is now not white European. And I always bring this up because a lot of people just kind of follow the best practice pipeline, but you have to be really mindful of what your actual question is and how it's going to affect your data, right? Because let's say I'm looking at cardiovascular health in African Americans in Detroit, right? I don't want to go to a typical GWA reference. I want to find one that's more appropriate to my population, so I actually capture that novel variation I'm looking for. And also, another caveat is this really interesting paper came out this year that, or not this year, I guess a couple of years ago, that actually suggested this VQSR is not really necessary, and that your kind of raw VCF files after variant calling are the same quality, if not better, right? Oh, and as I mentioned, depending on what you align your genome to, for instance, there's human genome 19 or human genome 38 are kind of the two standards. Your position and your ID are going to change. So when you open your VCF file, you need to maybe spot check a few of these and see what genome it was aligned to, because you can't merge VCF files that were aligned to different genomes. You'll have a lot of problems downstream. Oh, thank you, Candy. So like I say, my biggest takeaway for kind of the background today is know where your VCF files are coming from and be aware of what the best practices are, but also be aware of what the best practices are for you and your particular research question. And if you want to learn more, there actually is a workshop here at the collaboratory where they go over variant calling. It's with Dr. June Zhang, who is lovely and really nice, so I highly recommend that. Okay, so now we are going to get into more of our hands-on exercise for today. So there's a lot of different tools to QC VCF files, right? I personally use a combination of these. I'm sure there's more. If anyone wants to like throw in the chat other tools they've ever used. So like for instance, I'll do my merging with BCF tools, or I'll zip files with BCF tools. And then I'll run my statistics with Plink. So after this class, I would highly recommend checking out a lot of these different programs. But for today, this is a Plink workshop. So we're gonna talk about Plink. And Plink is really awesome because it's really fast, especially when you run it locally on your computer and you're not working with huge data files. It has a ton of different functions. So if you type in, oh, actually, you know what? So you're using PC, right? Like a PC. Yeah. Is anyone on Zoom using a PC as well? Or is it all Macs?
I'm on Windows.
Windows, okay.
Me too.
Okay, so we do have a couple, great. So I am not as familiar with PC. Dan, you might have to let me know what the appropriate programs are, but I'm gonna try and mention both as I go along, like what's good for PC and what's good for Mac. But so you basically, you can pull up your terminal, you can pull up your command prompt, I think it's called in PC, right? Command prompt, yeah, excellent. And you can do Plink and then this double flag help, and it'll list a ton of the commands that you can do in Plink. And Plink also has this really wonderful manual online where, compared to a lot of different programs, they go in depth on what all these different functions do and what you can do with them, basically. Okay, sorry, I see there's questions in the chat. PC, Mac. Okay, great. So we have a good mix, looks like. I'm gonna try and keep the chat up. Okay, so did everyone, oh wait, we didn't. Okay, so now we're going to download Plink, okay? This is the website. It's pretty easy to type. So be sure to click on downloading Windows or Mac. And today we're gonna download Plink 1.9. Okay, so I am going to show you how I would do it. If anyone on Zoom is confused. So it's www.kinship.org. cog/genomics.org/pink/9. So I'll put it in the chat. And then you're gonna just download. It's really fast.
It's like it's a zip file.
It's a zip file, exactly. So you're just gonna click on it. You're gonna unzip it. Okay, and then you're gonna have these different functions. So for this workshop, I want you to drag these different files into whatever folder you want to work with, right? So this program has to be in the folder with the VCF files, okay? But in the future, if you're comfortable with using like Conda or downloading something locally, you can just download Plink to your computer. Otherwise, we're gonna call up this program from this file right here, okay? So like for instance, I have my day one folder. I have all the files, right? I'm just going to literally drag them in this file, okay? Everyone good on Zoom? So the reason we are using Plink 1, good. Thanks for the thumbs up, Kristen. Everyone else, hopefully good. So the reason we're using Plink 1 is actually Plink 2. Oh, great, more thumbs up. If anyone has a thumbs down, please get my attention somehow and I can help you. So Plink 2 is the one that they're updating right now on the website, and they actually did an update last week and they forgot to put the appropriate like domain link in there. So if you click to download Plink 2, I don't know if they've updated it, it takes you to a dead link. So in the future, I highly recommend also downloading Plink 2, but you know, life, right? They don't have it right now. So Plink 2 is important because it preserves that phasing data. So if you have a VCF file where the haplotypes are already phased and you want to keep that, right, for your later analyses, you're gonna wanna use Plink 2. I have made the mistake many times of going through my entire pipeline, not thinking about it, using Plink 1, and I've lost all that data. So you basically have to start over with Plink 2, with your original VCF file. So please learn from my mistakes. However, a lot of people still use Plink 1.9. I would even say it's more popular because it still has a lot more functions than Plink 2. But you can find most of what you need on Plink 2. Okay, so if you haven't already, again, please download all of those data files on the Google Drive. This is a really long, ugly link, so I recommend, I will put it in the chat, but it should be in your email as well. So download those and put them in the same folder, like so. Okay, and then the last thing we're gonna do before we take a little break is I highly recommend downloading a text editor. It's gonna be really hard to follow along with what we're doing if you don't have a program that can open the various Plink files we're gonna be working with. Microsoft Word won't work. Excel's kind of finicky, so I use either BB Edit for a Mac or a Notepad, right? So I can put those in here. Okay, so now let's take a 10-minute break. So we'll come back at 10 a.m. Get coffee, whatever you need. Notepad should be fine. Like I said, I'm not as familiar with PC, so I was just kind of Googling what the standard is, but if Notepad is downloaded on your computer, I think that should be fine. And in this 10-minute break, if you've had any problems with downloading things, please let me know now, and we'll be back. Okay. And you had problem, right?
Yeah. So this is what comes out of it.
Okay.
This one.
Oh, gotcha.
But I don't know if it's
Right. Do you have the data files in a folder? Just try dragging it to Notepad and see if it'll open it.
from the one.
Yeah.
Okay.
So try and drag the head file. That one. Okay, that seems to be it. And then maybe you can try and drag like a VCF too. I usually drag it to the program on the Docker. I usually drag it to like program, so it'll open multiple files at once. Do you have it like on your desktop? Yeah. No. Okay. So maybe you could do like a left click and then open this. Okay. For a cohort. I just want to make sure. Yeah. Okay. It seems like that should be fine. Okay. Yeah. Okay, if Dan says Notepad++ is better, then maybe try that just while we have a break. Or was that not downloading?
I downloaded it, but I just couldn't find it. Could you understand like how I downloaded but I couldn't find it?
Yeah, one second. So Kristen, we're gonna go through how to call Plink, but basically the difference is you are calling it from a folder. You are not calling it from being downloaded on your computer. So the first thing you're gonna do is you're gonna change directory and then so you're gonna change directory and then you're gonna whatever your folder is called, I drag it.
Okay, I did change directory into the day one folder with Plink.
So that's the first step. And then what's very important, which we'll get into, so everyone else don't worry about, is you have to call it with this dot and this dash and then Plink.
Dot dash. Oh, okay, thank you.
Okay.
Thanks.
Maybe in yeah, so try that. I wonder if you have to, oh, there it is. See, that's it. Yeah, do that again, and then maybe it'll come up. There we go. So in the future, what I do is like, now that you kind of have it open, Yeah, I just want to move it to the right a little bit. So then you can like try and drag another file to that. Yeah, so try and drag like the second ACF file, the cohort 2, drag it there. See, now you can have multiple windows that yeah, it's a little easier than dragging it, like going through all that stuff. Yeah. Yes.
This is a quick question. I want anybody to move very quick from it. So like, I will get a little reference.
Okay. Yeah. Yeah. Do you want like a Popline book on the theory or on the okay. I've read that one before. That one's pretty good. There's one by Doc Edge that's pretty good. Shit, I'm sorry. Maybe just text that or not text that, write that and then put, I don't remember his first name. It's Mike. And then it's Doc Edge. Yeah, it's his nickname. I can't remember his name. I'll have to look for it for you. Yeah, that's totally great. You can download Plink locally to your computer. The examples I'll be doing will be with the .dash Plink. But if you download it to your computer, you wouldn't do the .dash, you would just write Plink. So it's just a very subtle change. But if you're going to be using Plink a lot, yeah, it's definitely tedious to make sure that it's always in the right folder. So it's just better to have it installed on your computer. So I feel like there should be a plug, right? Since my desktop is a little chaotic, I'm going to move some stuff because this is weird. There we go. Okay. We are going to zip right along so we can hopefully finish a little before lunch. I should say before noon. So a lot of different doodads going on. Trying to Can everyone on Zoom see this screen okay? I'm thinking it might be better to keep it down so I can do the steps with you.
Yeah, I can see the screen.
Okay, great.
Yeah.
Okay, so did everyone give me a thumbs up? Everyone got Plink okay? Everyone got the files okay? Excellent, thank you so much. Great. Okay, so for our first exercise, we are going to be pretending that, oh, you guys are in the way. We're going to be pretending that your boss has asked you to analyze case control data. So you've been given 2 VCF files of 2 different cohorts, and eventually we're going to use that data to basically look for a variant related to cancer, right? So the first thing you need to do is to take a look at these files and then eventually merge them so that you can do analysis later. And we're going to be doing the actual analysis on day 3, but for today, we'll take a peek and then merge the data sets. So what you're going to do is you're going to open your terminal or your command prompt, and then we're going to first change our working directory to wherever everything is, and then we're going to copy, paste, or type in these commands. So all of this script is in that day 1 folder. It's called the day 1 scripts if you want to copy paste, right? Because this is really tedious to type out. And it's a little hard for me to put it in the chat each time. So I highly recommend doing that, just having that text file open so you can just quickly copy paste. But I will do it with you. Why does this look weird?
And the folder, yes, okay, day 1, got it.
Yeah, so you can call your folder whatever you want to call it, right? So for instance, I have workshop day 1. So I like to drag it. So I do change directory, CD, and then I drag it here. So you can type this out, right? This would be the path to your particular folder on your computer, whatever it is. And then you just press enter. No such file or directory. That is a lie.
I'm getting you also there. It says 2 openings.
So I would probably drag the folder to the desktop.
Also getting an error not recognized.
Okay, so I think that's because, let's try this. I think if you downloaded it directly and didn't change the name, it just doesn't like that there's too many words. So maybe just change that VCF Plink workshop to just workshop. Sometimes it doesn't like when there's too many spaces in the file name. Let's see if that works. I'm getting different error.
Oh, I think I deleted them.
Oh, that's because you're not. So your computer, I guess, doesn't like that there's spaces, so just call it day_1. Perfect. Okay. So for anyone on Zoom who's having the same problem, the people here were having a problem of, what was it, too many arguments or not found. So sometimes, for whatever reason, it doesn't like if the file names have spaces between words or dashes. Right, so we're not quite there yet, but we will get there. Yeah, so just rename your files or folders to something really simple. It's just kind of a quirk about your computer. How's everyone on Zoom doing? Did we call up the appropriate folder? So we're on the step open terminal and then call directory and then our folder name, right? So it should show that you're now in the folder. Great. Okay, so like I said, I'm going to be toggling between screens, so it's going to be hard to always keep this up. I can't hear you, Asha, unfortunately. Yes, perfect. Sarfaraz, that's a great suggestion. Yeah. Can you guys hear me? Thumbs up.
Yes, I think Asha isn't able to hear you.
Okay. Sorry, Asha, maybe check your microphone settings or your output settings, that sort of thing. I think it's working though. Okay, so now that we're in our working folder, the first thing we're gonna do is we're gonna convert our VCF files to a Plink friendly format, okay? So there's basically two Plink friendly formats. There's what's called a PED and a map file, and basically the binary version of that file, which is a BIM, a BAM, and a FAM. So we're going to work with both eventually over 3 days. But for now, we are going to output into a PED and a map file. So I just copy paste this command and it ran, right? So if I check my folder, cohort 1 VCF is now a PED file and a map file, okay? And if I say I wanted to take a peek at the original VCF, right? I could do head -5 VCF, and I could look at head output. Oh,
Dan,
1. Yeah?
do you understand, to see me more better, like, I can't call in that full at all. Maybe Dan has, like a Daniel, a changeling, and it's just not working.
Oh, so get rid of all the spaces. So just recall this folder workshop.
So we name it this.
Yeah.
Okay. So.
And then for day 1, put like an underscore between things. Underscore, but no space. So yeah, there's no space. There we go. So try try it now. Oh, so you need to put CD.
CD.
And then space. And then I drag it. And then
CD space.
okay, so it says instead of a CD command, you can use a push key. So the word push.
Space.
No, E, space. Excellent. There we go. Okay. Interesting. Thanks for sharing. So, like I said, I'm not as familiar with PCs, so please definitely share if you find that anything needs to be adjusted for PC. Okay, so what I was doing before is say your VCF file is really large and you want to take, yes, thank you. You want to take a peek at maybe the head of it. You can say head, this flags the first 5 lines, the VCF file name, output, and then I just called it head output, right? So then I can take a peek. And this doesn't tell me anything, right? This is just my metadata. So maybe I want to see, let's say the first 50, right? Okay, so now I can start to get a peek at my VCF file. Obviously, it's going to be a little bit scrunched because it's in a text file, but you can see, all right, so I have unphased data. I have a few SNPs. I have that header we were talking about. I have GT, genotype data, that sort of thing. Great. So then we're going to finish with this, right? So we recoded our first VCF into a PED and a map file. Now I'm going to recode my second VCF into a PED and a map file. So I just copy paste. It's happy, right? It detects 100 variants and 35 filters. Okay, and the general format for coding in Plink is you call the program, right? So today I am showing you how to do it within that particular folder, but if you download it locally, or if you're using Windows, as mentioned, you would just write Plink, and then you flag the file format, you type the file name. Here we are keeping our allele order information, so I hit recode, and then output, it's outputting it into a Plink friendly format, right? So if I look at my folder, I now have cohort 2 and a map and a PED, and cohort 1 and a map and a PED. Perfect. And it's really important that you do the out, double flag, and a name. Otherwise, a lot of times Plink will overwrite your original file. So if I called it, so for instance, if this was a Plink file and I'm outputting a Plink file, here they're different file formats, so it's okay. But if it's the same file format, it'll overwrite the original one, so you'll lose your data. So good, just practice is to always give it a brand new name. Okay. So let's get some statistics on our new Plink files. So we're going to use this flag frequency. And again, you just copy and paste. and then it spits it out here in your folder. So I'm gonna look for it here. It is the .frq file. Okay, so then what I like to do, and you can do whatever works best for you, is I like to drag this file to my editor program, right? So I will call up BB Edit, and I just kind of keep it open to the left or at the bottom, whatever's best for you, and I just drag it in here. Okay, so it has now recoded our VCF file into a Plink friendly format, which is basically a glorified text file, right? So we have our chromosome, we have our SNP information, we have our allele, I believe, yeah, the first allele, the second allele, and then we have our minor allele frequency of the first allele. And then we have the number of chromosomes, which is the number of individuals that we see this marker observed in. Okay. So this is just this isn't doing anything to the data. This is just giving you a glimpse into the data. And what you would use this for, especially minor allele frequency, is when you go to remove bad data before doing analyses, right? So what we're gonna walk through today is how to look at all your data. And then at the very end, we're gonna basically cut everything we don't want. Okay, so now I'm gonna do that to my second cohort file. And I copied and pasted here. It spit it out. So if I wanted to, I can look at cohort 2 frequency, I drag it, and it gives me the same information. Okay. So now I have done it for you, but in the future, what you would want to do is look at that frequency file in more detail and compare the amount of markers between your merged data sets, right? So Plink can merge files with different markers, but it gets complicated. So I have given you 2 different files with different amounts of people, but the same markers. So Plink can very easily merge those 2 files. So what you want to do is you call it Plink, you tell it the first file you're going to be merging with. So now our flag has changed. It's no longer VCF, it's just file. And we're going to merge it. And then we tell it the file we want to merge it to. We're going to recode, so keep the allele information. And then we're just going to call our single merge file cohort. Okay. So as you can see, hopefully it's the same on your computer, but I've copied and pasted that here, and Plink is really fast, right? It spit it out almost instantly. So it's, even for bigger data sets, Plink is still relatively fast, which is why a lot of people use it. Okay, so I kind of wrote it here, but does anyone have any idea of, let's say I have like 10 VCF files or 10 PED files, right? The Plink friendly format. How would you merge multiple files?
Create a text file with details of the files and give that as the input.
Exactly, yeah. So you would basically create a text file listing all the files you want to merge, and then you would input that, the text file information here, and then you would merge it. So we're going to be doing something really similar with population information today. Okay, so when we look at our log file, right? So every time Plink does a command, it's going to output a log file, which basically tells you what command you ran. It'll tell you variant information. It'll tell you the amount of individuals. It'll give you like a timestamp if you want to see how long something ran for. It's really useful information. But if you open that log file, it'll look exactly like this, right? So we can see that from our first cohort file, we had 25 people. From our second cohort file, we had 35 people. Of these, the 35 people were new, right? They weren't in that first data set. And the first file had 100 variants. The second file also had 100 variants. Like I said, for today and this workshop, these variants are going to be exactly the same. And altogether, it merged these data sets. So now we have 60 people and 100 variants in our final cohort data set. And it also gives you genotyping rate, which is great. Okay, so we have a pretty straightforward example today, but merging files in general is really tricky, and you have to be really mindful of what your VCF file contains. Plink only handles the biallelic alleles, right? So if you have like, let's say a lot of copy number variants or a lot of alternative alleles, sometimes Plink can't merge those, so it'll discard them and it'll put it in an output file called missnip, right? And this is the names of all the variants that failed to merge between your data sets. So usually what you have to do is you have to open up this file, take a look at what variants failed, go in your VCF, see maybe why they failed, maybe try and figure out like what kind of variant they are, and then adjust accordingly, right? So it could be they're on different strands, right? They've been flipped. So then you try the flip option. It could be that there's a lot of like indels, right? So you probably just need to throw that particular variant out. It kind of just depends on your data set. So, in QC, it's a lot of troubleshooting, hence the merging files, it's a hassle. Okay, so when we do switch from our VCF file to our Plink format, we actually lose information, right? We lose that covariate information that is in our map file. So let's say, let me explain better. Let's say that your boss gives you a PED and a map file, right? So it's already a Plink friendly file format. That map file is gonna have your covariate information. It's gonna tell you like the age and the sex and any other data on those individuals. But if you start with a VCF file and you change it to a Plink file, a VCF file doesn't store covariate information. So therefore your new PED and map file aren't going to have covariate information. So it's important to keep that information in a separate text file. So for us, that is research_covariate text, right? So we're going to be using that on the third day when we look at regressions. We'll need all that covariate information. But always keep that in mind, because that's probably not going to be in your PED file if you're converting from a VCF. So the headline is lost after the merge, just because that's just the way the Plink file format works, right? It's just basically a plain text file. So you lose a lot of that first few lines of meta information data that was in a VCF file. Good question. Okay, so we're just gonna do one other function that I think might be useful for you guys. Let's say you already have an idea of the gene or the SNPs you want to look at. So you just want to extract from your files that particular region, right? You do the same thing. So B file is if we have that bed, BIM, FAM format. We have a PED and a map file. So I'm going to change that to file. So Plink file cohort. I put these SNPs, and and then output cohort SNPs. So make bed basically converts it to that binary head file. So you can keep it, not keep it, it's really personal preference. Okay. So I ran this and I extracted my particular SNPs. They are now in a binary Plink format file. So bed, bim, fam. So, did this open in here? No. So it's easier to see PED files in a text editor. So I'm going to rerun it and I'm going to output with PED. Interesting. Okay, so it looks like when you filter, you need a make bed. BIM file. Okay, so if we look at our BIM file, we can see I pulled those two SNPs and the particular location of those SNPs on the chromosomes, and then the allele information. So if you have a really big data set, probably more informative for our 100 people, it's just going to be pretty small information. Okay, so I've listed here, and I'll post a PowerPoint today, but here's some other file options that might be useful for you guys. If you want to look at particular genotypes for a particular variant, you would use this flag. If you need to troubleshoot and flip the strands, flip. If you want to merge multiple files, you would create a text file and then merge it with your original file, right? So these will all be up. Okay, so for our second and more pop gen exercise today, we are going to download a DCF file from the 1000 Genomes Project, okay? And like I said, I think this is really useful because I've seen a lot of people in medical studies who want to do a PCA of their individuals in a VCF file, but maybe they only have 50 or 100 people. And then those 100 people are like 4 different ancestries, right? So in that case, you're not really going to have a useful PCA. It's much better to merge it with public information so you can have a more meaningful data set. So I will put this in the chat. So everyone go to this link and we can download it together. Okay, so we're gonna do, it's kind of a confusing website. So we're gonna do it step by step. So after you go to that link, you're gonna scroll to the bottom. So it's gonna look like this. So we're gonna scroll to the bottom and we're gonna click on FTP site. So this is just one way to download these files. If you use Hoffman 2, you can download these files with Globus, which is a lot easier. You can also call it from your terminal, which is another thing I'll show. But we're gonna click on FTP. Right, and then from there, we're gonna click on data collections. So right here. And then we're gonna click on 1000 genomes 2504 high coverage. And if you're ever poking around this website and you're not quite sure what to download, my kind of starting point is I look at the last modified and I try and find the more recent files because I do update them. And then we're gonna click on working. So right here. And then we're going to click on the string of numbers, phase, SNV, indel. Okay, so hopefully you are all on this page. This is 1,000 genomes data across chromosomes that has been aligned to human genome 38. So human genome 38 is the more recent reference genome, so it's a little more updated. So just in case you ever need it in the future. So we are going to be working with chromosome 7. So go ahead and download chromosome 7, the VCF GZ file. That just means it's a zipped VCF file. And then download the index file. So the VCF GZ TBI. So you can just click on it and it'll download. Oops. Wow, the internet's really slow here. If you have Globus or you have Mac, the command I use is wget, and then you copy and paste this link right here. So FTP and then all of that. It's a little bit easier. Opening in 1 hour. Does everyone else have a really slow download speed, or is it downloading pretty quickly for everyone? Kind of slow.
It is slow, yes.
It's slow.
Mm-hmm.
Okay. Does yours say 1 hour too? Yikes. When I downloaded it, I'm sorry guys, when I downloaded it at home, it was like 4 minutes. Let me see. What we might do. Okay, so hopefully that is downloading for everyone. I think what we're gonna go ahead and do is we'll download the reference file and then we'll take a break and maybe we can do QC on a different file if that one takes too long. So the reference file is gonna be on this page right here. So I'll put it in the chat. We're gonna be downloading, chromosome 7. So maybe it's just the internet in this room that's really slow. Is everyone on Zoom? Is it downloading pretty fast or slow? I see one slow, one fast.
It's done.
It's done?
Yeah.
Okay, so go ahead. We're gonna take another break soon while everyone downloads. So go ahead and go to this link right here, and it's gonna take you to this page. There are way too many screens up. Okay, so if you want to know what's available on this site, you can filter, right, by all the different individuals they have and all the different projects they have. So the one we are doing is we're going to filter by data collection right here. And we're going to click on 1,000 genomes phase 3 release and then download the list. And this should be a pretty small file. Okay, so let's take a little break. And I have a USB, so for you guys here, you can kill your downloads and then I'll give you the files. Like we'll just transfer them to your computer real fast. Yeah, no, you're fine.
I got stuck completely, you know, like when my stuff hung and I was kind of behind. So we stopped.
Okay, so everyone on Zoom, we're gonna take a little 10-minute break. Let me know how it's going, if everything downloaded and everything like that.
Yeah, so we're able to troubleshoot it and was able to do this command. So
try and do just Plink. So instead of the dot dash, try just Plink. Just right, so like skip that dot and dash. Someone said for Windows, the dot dash wasn't working for them. So do the exact same thing. Yeah.
that space right.
So then, like, I don't know.
Like everything without the
Yeah.
Okay.
So they were saying this works for them on Windows. There you go.
Okay.
So you just have to. That's why it's like kind of tricky if you know your computer.
I never use this. Like this is first time using this.
Yeah, this is how you're gonna do Plink. Yeah.
What
Yeah.
the next step is? What's the next step?
You're gonna recode code. So do you have the data and scripts? So it's right here. Yeah. So now when you copy paste, just don't copy paste that dot dash.
Okay. So do this as well.
Mm-hmm. Yeah, let me get you the files, so those might take a minute to transfer.
Okay, so that copy. Okay.
And then what's the next step? Merging or frequency?
The next step I see here is let's get some simple statistics on the data.
Yeah.
So just code the way it says.
Yeah, and then after you copy the code, it's gonna output a .frq file
Into that folder.
Yeah, it'll output it in the same folder you're working with. Yeah.
The output folder is called how I identify.
So what does it say in your terminal? What would you call it? Yeah, so like this file, you'll want to open in that Notepad++, so you can take a peek at it.
That's it.
The .frq.
Okay. Well, so that's the output one.
Yeah.
Okay.
Wow, this internet sucks. Hmm?
I'm guessing there's this.
Yeah, I'm just because it was faster for me this time.
Yeah, it was so slow. So that would put here. So that would put here.
Mm-hmm.
The simple folder too.
Yeah, just to kind of get a, it doesn't do anything to it. It's just so you can
Visualize.
look at it.
Visualize.
Yes.
Okay.
Yes.
And do I do the simple
Yeah.
folder too?
Mm-hmm.
Let's see here.
Okay.
Yeah. So this will visualize.
Yeah.
And this will visualize what I'm looking for.
Mm-hmm.
visualize what I was looking for, just to see that it aligns with the color. Is that really what?
Yeah, it's just like kind of for your own personal benefit, so you can kind of look and see like what the frequencies are for
I
different
see.
variants.
I
If
see.
you have a specific question, really look at that.
Mm.
Yeah.
Okay, so just an eyeball test, really, nothing. Okay.
Yeah. It doesn't do anything to the file.
Kelsey,
Yeah.
are you connected to Eduroam Wi-Fi or are you connected?
No, I was gonna try that one though. I think it's faster.
Yeah, I think Eduroam is much faster than
Okay.
that one.
Do you guys want to try that too? It's EduRoam, EduRoam, and then you log in with your UCLA credentials. Hmm?
It went down to 4 minutes. The download.
Oh, it did?
It's so interesting.
Okay, so try UCLA Web and it's a lot faster, apparently. It goes from an hour to 4 minutes. And then, so you have the list. Okay, so it's working for you now, right? Excellent. I realize my USB is really small and I'm gonna do it. And then, so for the website, I'll walk you through the steps, okay? So go to www.internationalgenome.org/data.
Okay. And that's the design.
Mm-hmm. FTP site. I think it's data. Let me double check. I wrote it. Yeah, data collections. Yeah, so data collections, and then 1000 G 2504 high coverage, and then working, perfect, and then the face, and then yeah, download 7. So for everyone on Zoom, be sure to hang on to that merged cohort file because we're gonna work with it on Thursday. How's the download speed? It's already done? Excellent. I was wondering why it was so slow because I've downloaded it files here before. Okay, so 3 minutes. Yeah.
Do I need to know? I need to do that.
So the only thing you need to modify is just don't copy paste that. Yeah, that should work. And then this is just like, you don't have to run these are just for your personal references. Yes. So you can drag it. Yeah. And you called it cohort, so the head file. There you go. Yeah. So the reason it looks like this is what I was talking about is you lose your covariate information if you originally had it. So that -9 right here means missing information. That's your covariate information. But that's okay. We have our text file with our covariate information. So we have our individuals and we have all of our alleles.
How do you know if a branch in a problem?
It'll tell you.
Of the command project.
Yeah, it'll give you an error. Just like how you were getting errors with changing directory and stuff. Yeah. So I think briefly before we move on, I should very quickly show you. I can't remember if I did or not, so apologies if I did not. This was our merged head file, right? So we took those two VCFs, we converted them to Plink friendly formats, and then we merged it. So if you drag this here to your text editor, you can now see that you have your individuals, right? This is like the population level identifier. This is your individual level identifier. And this information is pulled from the alignment, right? Or your variant calling. So this information was built into the original sequencing. And then here we have our covariate information, and -9 means missing. And the reason it's missing, like I said, is VCF files don't store covariate information. So if you're given a VCF file, you're never going to have this information, which is why it's important to keep a separate text file with all of your covariates, because obviously it's going to be hard to run statistics without it, right? And then here we have all of our allele information, and the map file that it also spits out gives you the variant information, right? So we have our chromosome, we have our variant, and then we have our position. Okay, and then Plink is going to use those two files together to do different sorts of analyses. Okay, hopefully everyone has downloaded the VCF file. Yes?
So,
Oops, I still have, when I downloaded it, the time went up.
Oh.
It's back to like.
yeah.
5 minutes? Okay.
Yeah, it's getting back and forth. I mean, one of them is downloaded, I think, it's just one.
Yeah, the index file. Okay, is anyone on Zoom have any questions? We're just gonna wait. The Wi-Fi is a little slow here on campus, so we're gonna wait 4 or 5 more minutes for those VCF files to download. So if you have any questions, let me know.
I do. It's like, I think it-
Yeah.
they lost.
Oh yeah, so you're gonna click filter by data collection. And then we are doing 1,000 genomes phase 3 release. And you're gonna download the list.
I guess I should put this in my day one folder.
Yes. So Julianne, if you want to just call Plink, say in your terminal, you download it to your computer. I installed mine through a Conda environment. I'm not sure, Dan, if you have any other maybe suggestions for like PC, but if you want to call it, you would literally just write Plink. Yeah, so here's a resource on how to do that.
Yeah, Coda is also available on Windows. So
So, Dr. Jorgensen, my file is still downloading. I
Okay,
don't think it's going to finish within the hour.
that's okay. If it doesn't finish within the hour, just kind of watch what we're doing. I'm gonna post the PowerPoint, and all the steps we're gonna do should at home only take you like 5-10 minutes. Plink is very fast. So if you just want to follow along, that way when you do it yourself, it's easy. Okay.
Sure, that
Yeah.
works.
And then all the steps are going to be in the PowerPoint. But if you have any questions, definitely email me so you don't get lost tomorrow. So I think what we can do is, okay, how do I get rid of this? There we go. Okay, so while we're doing the last couple minutes of downloads, hey. Yeah, I just did it. Okay, so while the big VCF file is downloading, we are going to take a look at that smaller last file we downloaded. It's called IGSR_samples, and that one is the one that has all the population information. So what I like to do is I take it from my downloads, right, and just move it into your working directory so you don't lose it. So it's going to be called this, right? It has .tsv. Hopefully that downloaded for everyone. It's a pretty small file. And I personally like to open it in Excel. Oh, yeah, go ahead. Do you have a question?
Yeah, I was wondering where to download the file that you just showed, the TSV file from.
Yeah, so I think it should be in the chat. It is this right here. This link. Oh. You're gonna do filter by data collection and then click on 1,000 genomes phase 3 release and then download the list. So this link in the chat.
Filter by data collection and then 1,000 genomes phase 3 release, you said, right?
Mm-hmm. Yeah, and then the button download the list.
Okay, got it. Thank you.
Yeah, no problem. Okay, so you guys can take a peek at this, right? This might be interesting for you in the future because there's like, I think 3,000 people included on 1,000 genomes, and they're from all over the world, right? So there's lots of really interesting things you can do with this data. Here we go. And what we're gonna do is we're gonna create a text file and we're gonna extract populations from our big VCF file, right? So the big VCF file that's taking a long time to download has all of these people in it. It has 3,000 people. We're gonna work with like 300. So in order to create our text file, right, pull this up. And what I like to do is I go in Excel and I select it all. And then I do data, sort, and I sort by this column, which is population code. So my list has headers, population code. Okay. And then it'll resort, and you can see what these codes stand for if you play around with it. So like ACB is African Caribbean, BEB is Bengali, that sort of thing. And the populations we're going to be working with are CHB, which is Han Chinese, FIN, Finnish, MXL, Mexicans living here in LA, and YRI, which is Yoruba. So let's take a few minutes to do this, but we're going to copy and paste this first column. So just scroll down. I'll put this in the chat. Oops. These are the populations we want. So you can do like a Control F. And then I just copy and paste these names. So copy, paste. And then I make a new text file. Actually, you know what? It's probably easier in a new Excel. So I make a new Excel and I just do that. Okay. So we're gonna repeat that for the 3 other populations. So I'm gonna get CHB. Copy, paste, and I just stick it under. And then I'm gonna get finish, FIN. Copy, paste. And then lastly, Yoruba. So copy, paste. Okay. How's everyone on Zoom doing? That okay? Thumbs up? Cool. Okay, so if you recall, when we looked at our head files, right? So we made our merged head file. Plink needs 2 header lines, right? It needs a population level line and it needs an individual line. So when we extract certain individuals from a Plink file, we need 2 lines like this. And 1,000 genomes doesn't have different lines. So what I do is I copy and paste this exact same column and I just duplicate it. Okay. So it should look exactly like this. And then I do file, save as. If you do this in a text editor, you could just do file, save. And I called it population list txt. and I'm saving it to my working directory. Oh, okay. It doesn't like it because I have 2 windows open in Excel. So I'm just going to copy paste into my text editor and then save it there. Population list. Workshop. Save. And I already have one. That's okay. How are you guys doing?
Might have to download this one for
Oh, so we're not using the VCF file, we're using the smaller,
the Excel.
the TSV
Okay.
name file.
So,
So, oh yeah, no worries. So go to your downloads or to wherever you use that. So I think you already moved it to your work folder, right? This one. So we were just playing with that one. Perfect. So then you're just gonna go for those 4 populations and copy and paste that. So the 4 populations are really having trouble with the so these are the 4 populations we want to copy and paste this header information for. You're having trouble downloading the TSV file?
Yeah, when I click on download the list, it just opens the list.
I have a solution to that.
Oh yeah, what's
When I did it in Safari, that
gonna say it in
happened.
the no. So you want to look for those 4 populations.
That happened to me as well, but when I switched, I tried doing it I switched to Chrome then, and then it downloaded properly.
I see. Okay. Let me try that. Thank you.
Perfect. Thank you so much. So I select the whole thing and then I do data. Yeah, and then data, and then sort. And then sort. Oh, my data has headers, so and then sort by, and then population code. And then filter. So then you're gonna look for those 4 codes and copy and paste them. So I do like Control F. So there you go. So then you're gonna copy and paste to the bottom of those.
That worked. Thank you.
Excellent. Yay. Group troubleshooting.
Everything, like the entire.
Nope, just the first column.
Oh, just the column.
And then I drag it. I'm gonna I'll write down the populations for you. And then you can, that way I can. Yeah, and then you can copy and paste it into a new Excel or into a text editor. Oh, so you're too far. You don't want to see it. Yeah, so these are the 4. So you want to do the other 3 populations and like put them all in one column.
But I should save it, right?
You can save as you want.
What do you call it?
I just called it population list.
Oh, I see.
But you want them all four in that 2 duplicate columns. It might be a little easier for you to put it in Excel like this. And then like I copy and paste the 1 column and then I duplicate it. Yeah. Okay. How's everyone on Zoom doing? Any other questions? Is this looking good for you guys?
Does the order matter when you copy and pasting those calculation code?
I'm sorry, I didn't hear the first part. What is better?
Does the order matter? Like when you pasting like CHP, bin, MXL, YRI? Does the order matter or?
No, it does. That's a great question. It does not matter when you're extracting, but it matters if you think back to the example of, say, you are merging multiple files and you're creating a text file like this. You will keep the output file that tells you the order, because later on when we go to do a PCA or admixture plot tomorrow, you have to know the order in order to put the ancestries in correctly. So for this stage, it doesn't matter, but it's important later.
Okay, I see.
Yeah.
Thank you.
Uh-huh. Okay. So don't worry if you're still copy pasting. We're just gonna do the next step and then look at the output file for a minute. So here is the next step. Keep in mind if you guys need to get rid of that dot dash or not. But basically we are calling Plink, right? We're back in our working folder. We're calling Plink. Oh, I'm sorry. For that big VCF file that downloaded, I personally rename it because it's a really long, ugly name. So I called it all pops, all underscore pops. Because I personally don't really want to type this out every single time. So that's why it's renamed in my command. So I recommend taking maybe a second to call this VCF file whatever you want to call it. That's a little more manageable of a file name. So just to go over the command, we're calling Plink. We're telling it it's a VCF file because we're working with a new population file. And then I renamed it to a shorter name, all underscore pops. And then I want to keep the populations in this text file we just created, the population list. And this flag is important. I want the allele orders to stay the same. And then I'm converting it this time not to a PED file, but to a binary PED file, the bim bam fam. And that fam file will tell us the order of the individuals. And then I'm outputting it and I am giving it a new name. I'm calling it chromosome 7. So if you have created your text file list and your VCF has downloaded, go ahead and run this command and then take a peek at the output file, right? And we'll meet up there in a minute. How's it going?
Yeah.
Good?
Do I have to do it in this order?
No, the order does not matter. Oh, so you don't need that. So what you want to do is these 4, you want to put them in one long column. Yeah, yeah. So if this is like one population and this is another, just copy and paste this and put it on there. Yes. So Kristen, I think what you're asking is, we downloaded that list. It's all the populations of 1,000 genomes, and I want to extract certain populations. So I need to create a text file for Plink to tell it what populations to extract. And to do that, I copy and paste the first column of the country codes. So
I think I just wasn't sure where the Excel file came from, because the smaller .vcf.gz.tbi.
It's no, sorry, that's the index file.
Oh.
It's the file called right here, you see it down here, IGSR_samples.tsv.
That was in the day one folder?
It's not in the day one folder. No, it is something you need to download. So it's a small file. So go to this link right here, the International Genome Data Portal sample. And then this is going to pop up. You're going to do filter by data collection, and then check this box, 1,000 genomes phase 3 release, and then download the list. And it should be pretty quick. It's pretty small.
You're on the first tab. Oops.
And if anyone is hasn't gotten to this step, don't be stressed out or anything. Like I said, I'm posting the PowerPoint slides tonight, and it has step by step of how to do all this. So you can do it at your own pace tonight if you're more comfortable with that. Okay. Oh.
Just a question.
Mm-hmm.
The step I just went through now.
Mm-hmm.
Kind of quickly, what was that called?
So that step that we just did, we are creating a text file to extract certain populations from a VCF file. So say you have a ginormous cohort of like 3,000 people, but you only want to look at a disease allele and maybe 100 of those people. So this is basically one method of how to tell Plink to extract those people.
And the column we extracted, these are like subject IDs?
Yeah.
Okay.
Yeah, exactly. And 1,000 genomes doesn't have like a population ID and a subject ID, so we just duplicate it. That's why we made it into 2 columns, because Plink needs the 2 columns. Yeah, great question. Yeah. Okay, so I'm gonna go ahead and run this command. So I have my VCF file that I renamed to a more manageable name. I have my population list text file we just created. So I'm going to go ahead and run that program, and it's going to take maybe a minute to run, right? Because there's like 3,000 individuals. So go ahead and run this if you haven't already. It's in your scripts. So Plink -- VCF all pops. Yeah. Can I see your VCF file? So it should be and just follow along, and I can stay after. I will also post it on. But I think, yeah, I think it just got cut off, so it doesn't have the information it needs. So go to this international genome. You got it? Okay, great. Okay, so for those of you that were able to run the code, take a peek at your log file, right? Or the output in terminal, and it should look like this, right? So we loaded our VCF file. It detected 3,202 people. It had over 4 million variants, right? And then after we do that population list extraction, we are left with the same amount of variants, but 477 people. Good? How's it going?
Yeah, so done with all the,
Uh-huh.
I'm done with all the instructions.
Great.
And I put it in Excel, I saved it into an Excel file.
Oh, so save it in a .txt file.
Yeah,
Yeah, and then go ahead and run this code from the day one script. Okay, so how are we doing on Zoom? The rest of the class, we're gonna be doing basic QC, which should be pretty straightforward. So congratulations, this is the hardest part, is downloading, well, not the hardest part. I would say second hardest part is downloading data and getting everything organized. So if you've gotten to this part with no problems, excellent. If you have any problems, let me know now before I move on. Oh, so we're past that? No. So just that. And then be sure to rename your VCF file.
To whatever it's.
Yeah, whatever the name is. So start with.
The name of the file will be from to this point.
No, so the flag tells Plink what file type it is. So you want to keep that, and then this is your file name, and then this is the extension. So, but don't copy and paste that file. Yeah. I think you copy and no, you can't. Yeah. So I would copy and paste and then delete it in the text editor. Yeah. Okay, so mine, I think took about a minute to run. It's pretty fast to extract those populations. No, so what errors? Let's look at your working directory and see what you have.
Oh, the one. Oh, I see.
So you need to rename it, right? So I thought this was a really long name, so I renamed it to all underscore docs. So don't delete the docs, just delete it. Yeah, it's just a really long name. I called it all. underscore box. And then you're gonna wanna do the same thing for the next one. Yep, excellent. Now it should run. Yep, and now it's ready. See it thanks in a minute. Are you still downloading? Okay, we'll come back. I'll be here, no worries.
Luckily, there's the scripts.
Yes, yes.
So we want the Excel like column like create into Excel, you know, then anything we have for now. We haven't done anything with Java.
You saved it out to a TXT file?
Yeah.
That's where it's pulling the information from. Yeah, so you don't need the Excel anymore, really.
Even though, this is just a policy question, even though I don't have the name of that file in the command, it's able to associate with that file. So I'm saying, like.
It does have when you saved
So it's
the individuals, you can export it as a TXT file.
So
So you want to duplicate it. Yeah, so it's probably going to try and run, but it'll abort. So you need to make this 2 columns.
same thing.
It might run with one, but and then file select. And so in the command prompt, we called it population list. So you want to export that in a TXT file. Yeah, so it's probably going to try and run, but it may kill itself. Okay, so I just have a few slides on like QC in general. So basically, what is QC, right? And why is it so important? And what we kind of went over before is that pre-variant calling QC. So when the raw files are sequenced and aligned to a genome, we have a lot of the quality and filter information here. And a lot of this comes from Illumina codes, so you can always look that up. And when we think about after we variant call, right, we still want to do another quality control step. We still want to make sure that what we're actually calling is good for our later analyses. So pretty straightforward. And we can do all of this QC with Plink, right? And there's a lot of different QC assessments. There's getting rid of missing data, getting rid of incomplete data. We can get a lot of filters and reports on genotyping, allele frequencies, Hardy-Weinberg. And we can look at individual heterozygosity and kinship. And so for today, we are actually going to skip doing kinship because, like I said, Plink 2 is down. And you can do kinship, you can do IBD, identity by descent information with Plink 1, but it's kind of complicated and it's not the most straightforward method, versus Plink 2 does kinship estimations or kinship coefficient very easily, and the output is really easy to interpret. So like I said, Plink 2 is down, so I did this for us. So basically what you do is you run, I think I put it in the script, but if not, I'll add it. You run Plink 2, oh, maybe it's here. I inputted our file right with the extracted populations. I made a table of the kinship coefficients. I put that table in Excel, and then basically I just sort from largest to smallest. So as you can see, if 0.354 indicates you have a duplicate individual or twins, that's really high kinship. And we want to remove those people before analyses, because if you have duplications or you have people that are very closely related, this could lead to false simulations of significant data. Of course, if you're doing a medical study of families, this is not a step you want to do. So just be mindful of what your research question is. But what I do is I run that code, I look at the kinship column and I sort, and then I basically recreate that whole process we just did. Oh, sorry, this is what it looks like with 1.9. It's not as intuitive because then you have to interpret this. Yeah.
The kinship column would be every subgenetic data we want to analyze.
So
with?
It does not come with. Plink calculates it for you.
Okay.
Yeah. So the question was, does the kinship information come with the VCF file? And no, it does not. This is something that you can calculate with Plink. Yeah. So basically I recreate that whole process. I create a text file of individuals that I want to keep, right? So for my own research, I want to remove first degree relatives. So I use as a threshold point 0.177, and I take all the individuals below 0.177, I throw them in a new text file, and then I run that same Plink code we just did, right? I extract from this population the individuals who are not related to each other. And
Kelsey,
yeah.
does that value change with respect to population size, or is it like always that threshold?
Yeah, that's a great question. So it doesn't really change according to population size, but it does change depending on your personal research preference. So I've seen some people who do stringent thresholds of 0.5, and then I've seen some people who do lower thresholds of like 0.277. So it kind of depends on what your study is and how many individuals you have. So I guess it's like kind of related to population, but say for instance, if I were to rerun kinship with just a single population. So here we have Mexicans, we have Yorubans, we have Finnish, and we have, who am I forgetting? Han Chinese. If I were to run this kinship coefficient for individual populations, it wouldn't change that much because the individuals within the population are the ones who are going to be the most related to each other. Does that make sense?
I see. Thanks.
So this is kind of like a general standard, but you could go up or down depending on how stringent you need it to be. But for us, I did first degree relatives I wanted out of our data set. So I repeated the whole process and I filtered it. And now if you look back to your day one files, we're gonna pretend like we did this step together, and we're gonna take off from these chromosome 7 _kin files, okay? So everyone look back at your day one folder, and we're gonna do the next steps together with these 3 files. So make sure they're in your working folder. So if you're feeling really ambitious and you wanna do this process on your own, you can take a look back at these slides. Here's how I would do it in Plink 1.9, it's in the manual online, and then Plink 2 is how I would do it here. Okay. So hopefully everyone has these files, and we're gonna run some quick QC metrics on our filtered files. And then the very last step we'll do today is we're gonna remove all of the stuff we don't want from these Plink files. So the metrics we're gonna run right now do not alter these files. They just kind of give us like a glimpse into them. So for our first metric we're gonna run, it should look familiar. We did it once before. We're gonna look at allele frequencies. So you're gonna put in terminal Plink. Now we're working with a binary PED file, so it's flag B file, and we're working with that filtered kinship file, so it's chromosome 7_kin. We're gonna flag frequency, and then this is what our output is gonna be, right? So I will put this in the chat. And run it here. And as you can see, it calculated frequency pretty fast. So now I'm gonna look for so now it's chromosome 7. Okay, so now it's gonna be chromosome 7_frequency.frq. This is gonna tell us the frequencies of our different variants. So I drag it into my text editor and I can take a peek, right? So it's gonna look like this. Again, the chromosome, the position, our 2 alleles. Now we can see there's some variation in there, right? Maybe we see some deletions and insertions, our minor allele frequency, and our number of chromosomes per individual. And what's interesting about this is we have some zeros, right? It wasn't able to calculate the minor allele frequency. So we're probably gonna want to filter out those individuals later. Or I'm sorry, the opposite. We're gonna wanna filter out high minor allele frequency. So our next QC marker, did you guys find those files okay? Yeah, so they'll be in the day 1 folder. So it's gonna be chromosome_kin. Yeah, so you can do all these steps with us because we're pretending like we did kinship together. But you don't need the file you're extracting the population information from. That's just so you guys can practice like getting it. Okay, so the second QC measurement we're gonna do is Hardy-Weinberg disequilibrium. And sorry, I went too far. Hardy-Weinberg disequilibrium, and basically it's a kind of theoretical principle that states that genetic variation within a population is constant, with the giant caveat that there are no external forces that influence it, right? So if you have genetic drift, if you have mutations, if you have natural selection, if you have non-random mating, this will all throw Hardy-Weinberg out of whack. So Hardy-Weinberg it rarely applies in reality. It's kind of an idealized state for a population. So again, depending on what your research question is, you may want to skip this step. I usually skip this step because I'm looking for novel variants that would be affected by these forces. So filtering for Hardy-Weinberg would remove the variants I'm looking for. But if you're looking for common variants in a medical cohort, you would definitely want to do Hardy-Weinberg. And you can change the threshold. The typical threshold is 0.001, which is the default. And then this is the Plink command that you would run to check Hardy-Weinberg in your file. Okay, so copy and paste that in your command line. And it's gonna output a file ending in extension .hwe. And I like to open that in my text editor, so you can get a peek. Again, we have the chromosome, we have the SNP position, the different alleles, the genotype rate. We have observed versus expected heterozygosity, and then we have the p-value. So if our threshold is 0.001, how many SNPs defeat that expectation of being in Hardy-Weinberg, right? There's quite a few that are 1. And if you were to apply this filter, it would throw out those variants. How are we doing on Zoom? Thumbs up? Any questions? Great. Okay, so then a filter that, oh, sorry.
Is the 0.001, is that a threshold? That's the threshold for the p-value. So if it's
Exactly.
so it's filtering anything with a p-value. Okay.
Exactly, but we have not filtered anything yet. These commands are simply to look at these quality control assessments.
Okay, thank you.
Yeah, so, and I'm a very visual person. I always like to look at everything before I filter, because I just want to get a glimpse of like how many of my variants will make it and how many will be filtered out. So I'm showing all of you this. If you're also visual people and you want to kind of peek at your data before filtering.
Is there a reason why we're using such a stringent p-value cutoff instead of typical 0.05?
So it's not p-value in the way you're thinking of just like, is something significant? It's like p-value for this state of Hardy-Weinberg. I'll have to think on it and maybe give you a better explanation tomorrow. But what I've always seen is if you expect that a population is in this idealized state and variation is not being affected from one generation to another, then this threshold will remove outlying variants that would throw off your data. So it's not quite a p-value in the typical way you would think about it, I think. But I'll have a think on it and see if I can come up with a good example tomorrow.
Thank you.
Yeah. And then a big one that you probably always want to do, no matter what type of data you're working with, is eliminate missing data, right? So to get a peek at what sort of, did I put in the chat the Hardy-Weinberg? Yeah. To get a peek at what sort of missing data you have, you can do Plink, flag your file, double flag missing, and then output, and this will output 2 different files. It'll output a .imiss and a .lmiss. So the imiss is the per individual amount of missing, and then the lmiss is the per variant amount of missing. So you'll want to filter for both later on. And you can just drag these files with these extensions directly into your text editor to look at them.
Is mine correct? It looks like none of them are missing, or?
You are correct. Yes,
okay.
Definitely. So because we're pulling data from 1,000 genomes, they do a lot of like really stringent QC, and there shouldn't be any missing with our data here. But I have definitely, with a normal BCF, there's always something missing. Like you're always going to lose individuals and variants with this.
Okay.
But yeah, it should be all zeros here. So this is the the frequency that's missing. Okay, and then the last filter that we're gonna kind of QC for is outlying heterozygosity. And here is the command. And on a Mac, if you just kind of want to look at the distribution of the data, you can put these commands in. I think, Dan, correct me if I'm wrong, would these work for Windows too?
No, I don't think it works.
Okay,
It
sorry.
might be different command.
I think there's something equivalent. I'll try and look it up if you guys are interested. But basically this just gives you some numbers in terminal of what you're looking at. So if I wanted to look at the bottom distribution of heterozygosity, it just spits out numbers here. And if I wanted to look at the top distribution of heterozygosity, again, spits out some numbers. And this QC filter in particular is one that's really hard for me to visualize. So I always open it in Excel and I like to make a chart of it so I can actually look at the distribution of heterozygosity. So what I do is in your working folder, find the file that's .het, and then you add the extension .txt. And then I just open it in Excel. So I do like, okay, so I basically, instead of opening it in my text file, I opened it in Excel. And then I take this frequency column, which is the frequency of heterozygosity. And this is just a quick and dirty way. If you're comfortable with R or Python, you can definitely do that in there. And I just make a scatter plot, right? So like insert recommended chart and I click on scatter. And this kind of tells me what my data looks like across my individuals. So when I look at this, and what I have found is there's no hard definition of outlying heterozygosity. You have to look at your particular data and kind of draw a logical line of where to do the cutoff, right? So when I look at the distribution of our data, I see a pretty clear cutoff of some individuals I would remove. How about you guys? Any thoughts? So we have a nice clump here, right? They all seem pretty evenly distributed. We have a nice clump here. These seem pretty clumped. These are pretty clumped, but this one is a little more spread out. So I would probably cut these individuals so that it's a more even distribution across them.
Sorry, can you explain what the X and Y axes are?
Yeah, so the y-axis is the frequency, and then the x-axis is just, I think, the individuals. So it's the heterozygosity per individual for the variance.
Percent, okay, the overall percent heterozygosity for.
Yeah.
Okay, so someone from a consanguineous marriage, it would be like 0.5, you know.
Yeah, so as you can see from our people here, they kind of lie within this window of like 0.3 to maybe -1.5, right? So if I had an individual way up here in 0.5, that would be someone with outlying heterozygosity, and I would want to remove them from my data because they're going to throw the distribution off.
And what does negative versus positive on
Doesn't
the
really
Y?
anything for our purposes here.
Okay. Like reference versus alternate?
I don't know off the top of my head. I will
Okay.
I will get back to you tomorrow because we're gonna revisit this in the first part of tomorrow. So I will double check.
Thanks.
So this one isn't too bad, right? They're all pretty like clumped together. But if you wanted to be stringent, I would draw a line here. So these are just lines I threw on, right? As I said, they're all kind of falling within this distribution. So I would probably cut these individuals from my population. And those individuals, you can find them by, so here it's 0.3. So it's 0.3. I would sort by this column, largest to smallest, and then see what IDs pop up that are correlated with them. So there's 4 individuals that should have a frequency value of over 0.3. And then I would repeat the exact same code we did. I would extract all of those individuals but those 4 from my file using that same command of flag keep and then a text file and then the output. Okay, so now that we've kind of gotten visuals of all of our data, we are going to extract it. Or sorry, we're going to eliminate the low quality data. So all of those tests didn't do anything to our data, and this is the command that's going to remove poor markers as we define them. So here we have our file. I am doing a genotype flag of variants that are missing and more than 1%. I want removed. Mind means individuals that are missing more than 1% of their data markers. And Hardy-Weinberg, I'm doing that probability threshold of 0.001, and then I'm outputting. Were you raising your hand? Question? No, okay. How are you guys doing? Good? Okay? Okay, this is the last step. You're almost there. Okay, so how's everyone on Zoom doing? Did you remove low quality data? Great. So when you finish that, it should be pretty fast. You will get a log file like this, right? So we can see we loaded in a little over 4 million variants. We had 388 people. And after pruning, we got rid of our missing data. We got rid of those outside of Hardy-Weinberg. We got rid of low quality genotype data. Now we have, what is that? About 100,000 less markers. And a lot of these thresholds are kind of standard procedure, but you can adjust. Some people like to do 5% missing instead of 1% missing. Some people like to do 10%. It kind of just depends on how big your data set is. Okay, so give me a thumbs up when you're ready to move on. Got one, got two. Okay, so how are you guys? Yeah? Sticking in there? Okay, so, oh, I gotta check. Fail, file write failure. Can you show me what code you're doing, Meryl? Oh, and if you're doing a PC, copy and paste without that dot and dash.
That's all of them, but I will try without the.
Are you using a PC, Meryl?
Yes.
Okay, yeah, so in the chat, maybe try and copy and paste like this. That seemed to be working better with people with PC.
But now it says plink command not found.
Plink command not found?
Mm-hmm.
Okay. Let me go over these last couple slides, and then I'm gonna have you share your screen and we can go over it together, okay? Okay, so this is your takeaway for today. If you were able to run everything up until now, you may have noticed that we did not filter for heterozygosity in this command, right? But we definitely looked at it. So we saw that these individuals are falling outside of our distribution and we do want to remove them. So your takeaway for today is to look back at this Excel and identify the individuals falling outside of this limit, and then repeat that process we did earlier by creating a text file and keeping all the other individuals from this data set. And then save the log file for tomorrow and we'll pick up there. And then for those of you that are taking a grade, these are all gonna be part of your final assignment. So the other takeaway exercises are to identify a repository of VCF files that might be useful to you and copy and paste the link and maybe write a sentence or two about why they're useful to you. And then just take a look at the Plink website and the manual and kind of explore a little bit and pick out one function that might be useful to your research. Okay? Okay. So that's it for today. This was QC is always a beast. So we are through it, which is excellent. Tomorrow is going to be fun stuff. It's going to be my favorite things, PCA and admixture plots. And then Thursday is going to be GWA and regressions, and if you have time, CNVs. So we're going to use the data files we worked with today, and we're going to pick up tomorrow. Okay? So if you have questions, stick around. If not, I will see you tomorrow. Yes. Oh, yeah. And I will be posting after I go eat lunch. I'm going to be posting the PowerPoint online. So it's not going to be up immediately, but it'll be up in like a couple hours on the Google Drive. Okay. So Meryl, I'm going to stop recording. Do you guys know how I stop recording? There we go.